you for this introduction, and thank you for this invitation to this very cozy seminar. Okay, so I am Marie Sémon, and I am a professor at the Department of Biology at the ENS de Lyon, as Seth just said. And I'm also a PI at the Laboratory of Biology and Modeling of the Cell. But today I'm going to talk to you about a course we have launched a couple of years ago now, in 2016, which is entitled Practicals in Next Generation Sequencing. And I'm gonna try to convince you that programming course about high throughput sequencing can be fun, even in a generalist school for biology and even in a lockdown situation. So as I said, I'm working at the ENS de Lyon, which is a generalist school for biology. What do I mean by that? It means that the students we have here, which are represented by little figures like with different colors, they have to, when they arrive at the school, they can pick up from different themes, different teaching units that cover many of the fields that represent today's biology. So that some of the students are going to be choosing a very specific and specialized curriculum. This one in blue, for instance, is very interested in virology, so he's going to take a lot of teaching units related to virology. While others, that's the one that like the student in yellow, is going to be much more interested in plenty of things and exploring many topics: neuroscience, evolution, development, immunology. But all of them are going to take some statistic, even if they don't want to take statistics. That's because we think it's compulsory now and the importance of quantitative biology is growing. So we think that all students must grasp the basic methods. And some of our students, like the brown student here, will even become modelers. So they will take the statistics, but also programming and modeling courses. And if I try to represent where genomics is taught in our generalist school for biology, well, concepts and applications in genomics are pretty much everywhere, which are represented here by little stars. But most often, it's really shown as examples and as results, but the method to reach these results are often under the carpet, if you will, so that the methods in genomics for most of our students are very blurry and look like the picture on the bottom left, with plenty of lines, very complicated, and plenty of different colors. They don't really understand how it's drawn. So that's why we have, but on the top of that, we know all that high throughput sequencing, NGS, has become an everyday research tool, and so that it allows to address many questions. And our students have been aware of that. Yet, approaching the analysis step is scary for many of these students, especially if they are a little allergic to computers to start with. So we decided on a teaching progression from the bachelor to the master 2 level. And in the bachelor level, we teach through the command, the Galaxy web tool. Also, it doesn't involve any programming. But then we try to build up a skill up to the master 2 level, where they are able to do these practicals in NGS, where they really do their own programming. So this NGS analysis in blue here is a teaching unit I will tell you about today. And in this unit, we want the students to be able to discover the main methods in NGS analysis, also to discover and demystify command line and bioinformatics, because as I said, it's scary for many of them. And we also want them to discover good practice in data analysis, such as reproducibility, traceability, but I will come back to that in a minute. So, of course, this was a challenge to set up this teaching unit at the UNS school, but of course, I think also it's going to be a challenge for many other schools, because we face more or less the same difficulties. So, in this unit, we want the students to be able to analyze a data set from raw sequencing reads up to near publication figures. And as I said, the challenge is that we have students with a wide range of skills in genomics and programming to start with. So concretely, we put the students in team. So that's teamwork for 6 full days. And then we want them to come up with their own solutions. And the code they produce has to be reproducible and available on GitHub. This is all very simple in theory, but it's also very ambitious, of course, because we really want to help the students towards autonomy so that they can perform their own analysis afterwards. So we really try to coach the students without writing scripts for them, and that we handle through flipped classrooms, mutual aids between the students, and a lot of tutoring. So to attract the attention and to attract the interest of the students, we offer them a lot of topics for their analysis that would more or less match the broad interests they may have in biology. So that you can see in different colors. Some days, some years we would offer ecology, neuroscience, and immunology topics. Another year, evolution, neuroscience perhaps again, and bacteriology or cancer, etc. And among our students, also one maybe originality of this unit is that we will have typically 4 students per year that would be able to analyze the data and to prepare their own RNA-seq library or their own libraries and they do the sequencing and then they do the analysis. And the rest of the students would do a public reanalysis of a public data set. But that is very interesting as well, as I will tell you. So this is just examples of topics we have given to the students in the past years. So corresponding to the different thematics shown previously. For instance, in neuroscience, we studied data corresponding to a murine model of Alzheimer, but also we've been studying gut microbiota in Drosophila, genetic load in human polymorphism, adaptation to aridity in rodents. And I will detail 2 of them in the next slides, response to virus in bats and colors in clownfish. But of course, last year, like in many countries, we had to switch to remote teaching. And it was a bit challenging, like for everyone, I guess, especially for practical course like this. So we explored different possibilities and we found that in our hands, a simple Discord server was the best solution because it allows for independent rooms, so vocal and chat for the different projects, and because it's very versatile. It's also very good because the students are very they know very well this kind of platform, so it was very good to raise the team spirit because the students, some of them were quite low at this period. We also use this very simple camera to simply film the papers, to be able to scribble on the papers and replace the whiteboards or the blackboards. And that way we managed to set up a team working together, even if some of our students were working very, very remotely from France, here, for instance, in Peru. So some other technicalities, of course, we thought that it couldn't work if we didn't have any homogeneity between of the systems that the students were running. So we decided to rely on virtual machines that are provided in France by the Institut Français de Bioinformatique, called IFB. This allows to size the machines according to each project, and we were able to open 1 machine per student. And that way, the students were using RStudio servers that we deployed before the course. We all needed programs, so they don't need really to install new programs unless they really want to, which is still possible. And then that's very good because the students may use their own laptops if they wish, and then they connect via their web browser, but they don't need any special powerful machines. They don't need Linux installed. They just connect to the browser, and then they use the power of the virtual machines to run the analysis. I said that we installed the RStudio servers, which might be a bit surprising because it's not always used to program. But actually, it was quite convenient because it's a friendly programming environment. You have a graphic interface, which is good for students not familiar with command line. And also, you have a Git interface for versioning. And you can, of course, you can type R commands, but also you have a Unix terminal for just running Python or Bash scripts. And also it's very useful because the teachers can access the machines and correct the students if they are completely lost, which of course wasn't possible in a lockdown situation otherwise. So, as I said, I will detail slightly two of the topics to illustrate some of the main idea we want to convey with this course. So the first topic I would like to detail is the study of the response to virus in bats, which of course became very famous now. So this was a topic that allowed the students to prepare the data from the library, the RNA-seq library to the sequence, and then to the analysis. So more precisely, well, precisely if you wish, this topic was the idea that bats resist infections very well. We all know that by now. And interferons are secreted by the cells after virus infections, and they activate many genes. But we wondered what's so special in bats' interferon response. Maybe that's what allows them to resist infections. So here you have two illustrations of these concepts that bats resist infections very well. The bat we studied is called the Myotis vellifer. It's a model system of Lucie Etienne's lab in a neighboring lab, in the virology lab called CIRI at the ENS de Lyon. And as you can see, Myotis vellifer is laughing, even if it's surrounded by terrible pathogens, because this bat is resisting infections so well. I will show you, it's not drawings by Master 2 students, but younger kids. So, So the students, the Master 2 students this time, so I said they prepared and they sequenced the RNA-seq libraries, and it's a total of 3 days of work for them. And at the end of these 3 days, the libraries are ready for sequencing. So they treated, the cells, the bat cells were treated with interferons or not treated with interferons, and then you take the RNA and then you do the RNA-seq analysis, preparation. So that's done with in collaboration with a local sequencing platform in another institute nearby called the GFL, still at the ENS de Lyon, and it's with the help of Sandrine Hugues and Benjamin Gillet there. And then, so the students prepare that, and then we want them to come back to the rest of the class and to explain all of the steps of the library preparation to the rest of the class, including a detailed explanation of quality controls. This is a very simple example by a classical quality control multi-QC, but of course you have plenty of other quality control at the level of the wet lab preparation. And we really want the students to go thoroughly through these steps, to be really to criticize their own work, to be able to have some point of view of on the reason why some samples would fail or not later on. And then that's the 3 first days, and in the 3 remaining days, we want the students to find genes that respond to interferon, so in this bat, and to compare with homologs in humans. So that's on the right of the slide. You would see that these students have prepared 6 samples, 6 libraries, and then they took the RNA-seq. And you, among these 6 libraries, you have 3 controls, so without treatment, and 3 treated cells with interferon. And the students have been able to detect genes that are significant, that show different, significantly different expression between control and interferons. And then, obviously, they will pick up some of these genes and look for homologs in humans and try to interpret that with the help of Lucie Etienne's team in this particular case. Of course, we don't expect the students to make a controlled publishable analysis after 6 days, but it's already a rough first analysis. And I think it's interesting because it provides the local team with pilot data that often in the past has ended up in publications. So through this practical, we also try to we also tailor it so that it benefits local research teams, and because we really don't want sequence data to be wasted at the end of the practicals. And then with the same data, another group has studied the phylogeny of the genes reacting to interferon. So that is, these gene families in bats, do they undergo more duplications? Are they evolving more faster than in other species, and so on? So it's the same data, but the pipeline is, of course, much more complex. You can see a rough outline of this pipeline on the right of the slide. Of course, it's more complex pipeline, but it's also a very classical one, I would say. And we provide the students with a global backbone, but we want them to make the scripts by themselves. And we want them to understand the tools that they've been using. So again, we ask the students to read about the tool and then to be able to come back to the rest of the class and to explain how it works in a flipped classroom situation. So that's for the brief example about response to virus in bats. But now I will switch to just another example of another project about dataset reanalysis, and this time of colors in clownfish. So this was just from public data that the students would reanalyze during 6 days. So the point here is to reanalyze this published dataset from the raw sequencing reads, again, to near publishable figures. But then what happens most often is the following. Here you can see, so this example of this dataset was published by Salis et al. In 2019, and it's a classical RNA-seq experiment with, again, 6 samples, but this time it's 3 samples of this orange skin band in clownfish, and you have 3 other samples of the white skin band. And on the left, you see, so classically, when you have this kind of data set, you measure the expression of all genes in all of these 6 different conditions, and you put the table in a PCA analysis, in a principal component analysis, to see the main axis of variation. And what the students discovered this time is that on the first axis of this PCA, they find that they see well, it's not very clear here, but the samples more or less group according to individuals. So you have on the left these white dot and orange dot come from the same individual, and so on and so forth. So it's a very a genetic, or at least an individual linked difference. And on the second axis of the PCA, you have a difference between white and orange bands, which is what we are interested in this analysis. And the students were very disappointed. Why? Just because they didn't find the exact same pattern as what was published in 2019, which is on the right of the slides. You see that actually on the right of the slide, you separate very nicely the colors on the first axis of the PCA. And they are disappointed because at first the students, they think they made it wrong somehow because they don't find the same thing as the publication. Because for students, the publications are gold standards. And if they don't find the same thing, it's just that they had a mistake somehow. But then we go through the pipelines in all of these publications, and we figure that you always have differences, even small differences, maybe in the versions of the genome, maybe in the versions of the tools, or maybe it's not exactly the same normalization that was used for the normalizing RNA-seq data. And so, but sometimes you can see it in the material, in the methods of the paper. Sometimes in some papers, the materials, the methods are very reduced, and you cannot actually access the information. You don't see what was a version of this and of that. And so the students, by the difficult way, in a way, they get converted to reproducible research because they see that it's absolutely compulsory to absolutely mark everything you've been using to be able to reproduce a result. And so that's how we lead them to provide a code that is commented, that's with a versioning tool. So here you have an example of a GitHub provided by one of the students for another topic. We ask them to arrange their GitHub repository in a nice and organized manner so that others can reproduce their analysis. And sometimes, and this is another example from another topic, which was here a single-cell RNA-seq analysis last year. Sometimes it's not even possible to reproduce the results. And it's not because the authors didn't register everything they've been using, but it's because the data, the methods are evolving so fast, especially in single-cell analysis, that you need a dependency between packages that is nearly impossible to reproduce from scratch from another in another computer, because you have dependency between all of the methods you've been using. And the only solution for this is containerization, so Docker or Singularity or maybe others. We don't have time to teach that in practice for the students, but at least they see that it's necessary in some cases. And then we also spend some time to find reasons for discrepancies between published data and what they find, and that by using benchmark papers. So, and by comparing different tools. So in this case, with the single-cell RNA-seq data, the student was able to compare different mapping tools, but it's not always that they manage that. So to take a bit of a step back, I would like to tell you what I think about the pros and cons of this kind of teaching. So in comparison to classical teaching, I think it's very demanding for both the teachers and the students, yet we have a very good feedback over the years. I was afraid at first that less notions are taught than in traditional teaching. That's because each group of students is really doing just one example, and it doesn't see all of the different applications. So I was afraid of sprinkling knowledge, but actually it sticks better. And I think the flipped classroom works very well because they really understand better and they are less afraid to ask questions if it's a fellow student who is presenting. Also, I said that the students are working by groups, and we made heterogeneous levels between students, and it's done on purpose to be welcoming for beginners. And also, this teaching is demanding because also for the tutors. So often we have 2 to 3 PhD students in the course, and together we anticipate the questions and problems before the course, so that the tutors can handle their groups more independently afterwards. And I think that's a way to mentor young teachers so that later they can build up their own practicals or their own lecture more independently. So now, why do we think it works? Of course, we think it works, otherwise we wouldn't continue it since 2016. Because it's very, as I said in introduction, it's very ambitious. We want to make a full, even if rough, analysis in 6 days. We think it works because we have the EFB providing with virtual machines, which allow the corrections by the tutors. We think it works because we have many steps that are similar across projects and that can be shared between groups. For example, the quality controls may be similar and so on. And also, part of the mark is on the students' personal investment, so that for good students, this means helping the others and favoring collaborative behavior. And we think it works also because these are master 2 students and they are able to read benchmark papers. They are motivated because they have internships and they see their PhD coming. So they really want to understand and be able to do their own analysis. 2 last points. It works for the students can get a hands-on experience in library preparation and sequencing, and that's because our students at the ENS are already familiar with the wet lab techniques. And we have this nice collaboration with the local and motivated sequencing platform. And lastly, and I get this question often, it's very costly, both in terms of money and on time it's time consuming for tutors, but we really reuse a lot. We reuse the data. As I said, we use the data that will serve later as a pilot project for the local teams, local research teams. And we reuse the code and the projects for the tutors because we tailor the projects with the tutors to match their interests and their needs in research so that it can be part, in a way, of their PhD. So what's next? To conclude, so for the students, all students, we said that the contract, if you wish, was that all students must grasp the basic methods. And at the end, we hope that the students are not frightened by the command line anymore. And they will be now ready for collaborations or for running their own specific analysis. And we said that at the ENS, some students, and more actually, will become modelers. So we hope that it helps them to embark in a PhD in modeling. And some of them even try a second master in bioinformatics and in data analysis, because presumably they liked it. And on the same mode, we would like to open a new unit called Statistics in Ecology, and we will see how it goes. So, okay, I would like just to acknowledge plenty of people, notably my fantastic crew of tutors, mainly PhD students, as I said, Karine Ray, Romain Bulteau, Corentin Deschaux, Marie Cariou, Alice Hugues, and plenty of others since 2016. I really would like to thank the motivated students we have here at the ENS, the Institut Français Bioinformatique for providing the virtual machines, notably Christophe Blanchet, always super useful. The local sequencing platform, thank you Sandrine, Hugues and Benjamin Gillet. The Master Biosciences at the ENS de Lyon and the Department of Biology for letting us and supporting us in this adventure. And for the projects I have shown you, the BAT project, I would like to thank you, Lucie Etienne and Stéphanie Jacquet, the local research team in virology, and Franck Picard for his analysis on the SRNSEC analysis. Okay, so now I'd like to thank you for your attention, and I am done. Thank you.
That was a very good talk. Very interesting. So
hello? I can't hear you. I guess you can't see in the Q&A. But I had a question in the Q&A. can you elaborate more on why is it more demanding for trainers and students compared to in person? Oh no, I don't think it's more demanding to be. I don't think it's more demanding to be remote than to be in person. I think it's more demanding to have this flipped classroom thing and this not to write and to guide the practicals more. It's more demanding because we have to prepare at least 4 or 5 different topics so that each little group of students can work independently. And that's because we want really everyone to be involved. We want everyone to be not to rely on the others, to get the answers. So if you have 20 people doing the same thing, it's perhaps less motivating. And also we want everyone to have a topic that is tailored to his or her own personal interest in ecology or in virology and so on. So of course you have to prepare 5 times more. And also for the student, it's probably more demanding to be just in front of your computer, and then we ask you, okay, write a script now. It's more difficult than just giving you the script and then you run it.